Hi, everyone. My name is Kotb Abdelmohsen. We work at the NIH, National Institute on Aging, and National Institute of Health. And we have been working for more than 20 years on aging and age-related diseases with a focus on senescence and senescence-associated traits. Here today, we're going to discuss with you our work on the trophoblast stem cell secretome and its impact on cell senescence. I'm sure you're very familiar with this wheel, with this hallmark of aging. I call it personally the wheel of aging, and now for some investors, you can call it the wheel of fortune. You can spin and invest anywhere in this wheel, and you will get business. So basically, what I'm interested in, 2 points in this schematic of hallmark of aging. The first one is the stem cell exhaustion and the senescence phenotype. As we know, with age, stem cells, they get exhausted, meaning that they are not able to perform their function properly, meaning that the tissue will not be renewed or will not be repaired efficiently. That feeds in that the stem cells will not be able to repair the tissue because the stem cells, the senescent cells, as they accumulate with age, they keep secreting the SASP, as we heard in the previous talks. The SASP phenotype keeps aggravating the effect, or the negative effect on the stem cells, giving this kind of vicious circle here. They keep negatively influencing each other negatively. And this was one of the main thoughts that I have, that I would like to interfere with this kind of circle or interconnection between these two phenotypes in a hope that I can break the circle and to unlock the key for longevity. So, as we know, senescents were actually normal cells, and they get exposed to various stresses, as Maria explained yesterday. Telomere attrition, also we have mitochondrial dysfunction, and other stresses that would lead to the development of senescent cells. However, due to the decline in the immune recognition and due to the aggravation or the effect on immune senescence, the immune system is not able to clear the senescent cells effectively from the tissues. As a result, the stem cells keep accumulating in tissues as we age. Of course, this kind of accumulation goes up with age, and that would be involved in different diseases as we age. The hope is to come up with creative interventions like senolytics or senotherapeutics in a hope that we can overcome the negative effect of senescent cell accumulation in various tissues and organs. This is just a brief summary of the existing senescence-associated disorders or diseases. For example, we and other labs have found that neuronal cell senescence is associated with neurodegenerative diseases. Also, senescence is associated with cardiac fibrosis, lung fibrosis, and even the senescent adipocyte, they are involved in metabolic disorders. So this is just some examples, but it's really involved in various diseases, and we see them also in Alzheimer's disease, for example. We see them accumulate at the site of pathology. With that, this is one of my favorite slides because first time I heard this kind of pleiotropic effect of senescence was from the late Judy Campisi, and she liked to call this the pleiotropic effect of senescence, meaning that what was good in younger age turned to be bad in older age, meaning that when we were young, the senescent cells are actually beneficial. They suppress tumorigenesis, they help us in wound healing. However, when we get older, they do the opposite. They promote tumorigenesis, and you get inflammation and other age-related diseases. So how do we target senescence or try to combat the bad effect or detrimental effect of senescent cells? There are a few approaches that we're investigating at this point. So there are senolytic drugs, and these drugs mainly target the pro-survival pathway, BCL2, for example, in a hope that you can tip the balance more towards apoptotic pathways. And with that, you can induce the death of senescent cells, and that is known as senolysis, basically like lysing the cells. And common, like known drug is the combination of quercetin and dasatinib. They are commonly used now. The other approach is the xenomorphic drugs. Like as the previous speaker and the other speakers also during this wonderful meeting, if you inhibit mTOR or NF-κB, you can interfere with the pro-inflammatory pathways. And these drugs are known as xenomorphic or xenostatic because they basically don't kill senescent cells, but they stop their bad behavior and they stop them from secreting those nasty chemicals that promote inflammation. Other approaches like immunotherapy, CAR T cells were explained also. You can also target senescent cells with nanoparticle. This is targeted delivery system. And or you can also get some combination between genetic approaches and pharmacological approaches as well to target senescent cells. So this slide was really inspiring for me. Because once I saw the parabiosis is working and the old animal is really benefiting from connecting the circulating system with the young animals, this actually helped me to think about different approaches that I can use. For example, how about if I get the early stage of stem cells, secretome, and I can test it on senescent cells. And as you know, the secretome itself includes extracellular vesicles, or they are also known as exosomes. These are small particles. So the secretome itself can also include non-EV particles or soluble proteins as well. So I was deeply interested in testing this kind of idea. What happens if I take the secretome of stem cells, and how early can I go with the stem cells lineage so that I can test them on senescent cells? Just a brief introduction about the exosomes or the extracellular vesicles. These are membrane-enclosed nanoparticles. They are actually coming from the cells. As you can see here, the pathway is very well established. They are secreted by cells, and they are cargoes. They have proteins inside the vesicle. They have some DNA, RNA. They have small RNAs. They have microRNAs. They have a number of proteins as well. They have also even fragmented RNA, fragmented tRNA. And the idea actually is that when the components or the contents of extracellular vesicles, they move from one cell to another, they can influence the gene expression profile of the recipient cells. Common surface proteins are known on the surface of extracellular vesicles. These include basically the tetraspanin, CD9, CD63 that we always detect when we isolate exosomes. As you can see here, we managed to isolate the exosomes. Basically, this is the size distribution. And most of the small exosomes that we isolate, the range is between 30 to 100 nanometer. And these are the surface proteins that we commonly detect as markers of proper and good quality of isolation of exosomes. But the main question for me, actually, and for people who work at the NIH, two questions. How early can we go with this stem cell? What kind of stem cells are we getting? And also, one very important issue is how ethically correct it is. As you know, at the NIH, for example, we have not been able to work on a number of stem cells due to ethical conditions or ethical issues. And I did a lot of search, and I found that the company, Accelerated Bioscience, they have very early embryonic or stem cells that are obtained from ectopic pregnancy. And then these cells are isolated within after the fertilization between 4 and 8 weeks from the embryo. After as a result of it is ectopic pregnancy. This is very good for me, very good for NIH, ethically correct, because this is not going to be a viable embryo. And also, it turned to be within the early stages of embryonic development. Another point that is also within the moral aspects of NIH is saving the mother's life, because if this embryo or this ectopic pregnancy If it keeps growing in the fallopian tube, the mother will experience or suffer from internal bleeding, leading to cell to death. Eventually, I talked to the company, the Accelerated Bioscience, and I asked them, please give me all the media that you have so that I can test the effect on senescence. And then I started to design the project. As you can see here, we start by characterization of the conditioned media that they send, which includes the exosomes and other nanoparticles. And also want to test the effect on senescence. And eventually, we wish to go, or we're actually now are doing the studies on mice to test the effect of these nanoparticles or also the conditioned media on aging. OK, this is the first slide of the experiment. We exposed WI3A cells. These are primary human diploid fibroblasts. We expose them to ionizing radiation to induce senescence on day 1. And also, we incubate them with the conditioned media, either the control conditioned media or the conditioned media obtained from trophoblast stem cells. And we keep observing these cells like once or twice a day to see the morphological changes and maybe also the biochemical changes, as I explained next slides. As you can see here, we observed that the cells that are incubated with conditioned media from trophoblast stem cells, they actually get more confluent and they have more cells and they are morphologically different from the control cells, indicating that at this time point now we have to stop the experiment because we don't want to get into complexity or complications with increased confluency. So as you can see here, we see morphological changes, also increased number of cells as we incubate cells with the trophoblast stem cell-derived conditioned media. Also, we see an increase in MKI67, which is a marker for proliferation. This MKI67 is normally downregulated in senescent cells, indicating that these cells still have some capacity for proliferation. We also see a slight increase in lamin B1, which is also normally downregulated in senescent cells, indicating that the conditioned media actually can boost or give some promotion for the proliferation after exposure to ionizing radiation during the induction of senescence. As the previous speakers also alluded to the activity of NF-κB and DNA damage as well. So we observed that the cells that are exposed to the conditioned media from human trophoblast stem cells, they have less DNA damage. And as a consequence, the phosphorylation or the activity of NF-κB P67 is also less in cells that are treated with the stem cell conditioned media, indicating that what happened, or telling us what happened to the SASP factors that could be regulated by these master regulators due to the changes in gamma H2X and NF-κB. That led us to measure the levels of SASP, including mRNAs, including IL-8 and CXL1, GDF15, and other mRNAs that are encoding for secreted or SASP factors. As you can see here, at 4 days post-IR, we see a significant reduction in the secretome of senescent cells, the mRNAs that are encoding for the secretome. And also at 7 days, because these cytokines, they do have dynamics at the transcription level, we see a reduction also in day 7 in CXL1, IL-8, and IL-6 after the exposure after the treatment with the human trophoblast stem cells. I have to mention also that we also tested the same thing in other senescent systems, including etoposide, and we saw similar effects. Now coming to try to understand what is the mechanism that could be behind the effect of these conditioned media. And I like normally to do unbiased approaches. So we took the media, and we took the recipient cells, and we decided to do multiomics, or unbiased approaches. High-throughput cytokine array, bioplex, to detect the soluble proteins, or the SASP. We also perform mass spec analysis on the extracellular vesicles and particles in the media. We also performed some RNA-seq analysis, and we have ongoing analysis on the proteomics using Olink and the metabolomics as well. These high-throughput analyses would help us to understand or see, put our hands on some mediators or effectors that could be mediating the effect of the stem cell-derived media or the exosomes as well. So what you see here is the cytokine array showing you the cytokines that are, or the soluble factors that are in the conditioned media without incubation or without treatment or without being exposed to stem cells. And this is the conditioned media that we obtained from Accelerated Bioscience. These were the media that was used to culture the human trophoblast stem cells. As you can see, huge differences, and you can here appreciate that you can detect several proteins or several soluble proteins in the conditioned media obtained from stem cells. That would include IL-8, VEGF-A, and other factors as well. In addition to that, we performed the bioplex, and we could see some common factors between these two. However, this is kind of limited. You measure about 18 or from like 16 or 18 SASP factors in one reaction. But you can confirm here that IL-6, IL-8, or also MMP3, the all down upregulated, or the all secreted in the media that we analyze from stem cells. So collectively, we observe that these proteins or these soluble proteins in the conditioned media, we see that they are basically involved in many cellular processes, including cell proliferation, inflammation, immune response, angiogenesis, and also the extracellular matrix remodeling or tissue remodeling proteins. So this, we speculate or hypothesize that this rich composition can actually modulate different signaling pathways that influence different cellular processes that would include also SASP production. On the other hand, if we take the extracellular vesicles and particles that are released by the stem cells and perform mass spec analysis, we identify proteins that are basically involved in extracellular matrix, also cell proliferation and vesicles, and also we see different signaling pathways and cytoskeleton as well. We can also observe some immune effect on immune response, indicating again or confirming that the media is actually very rich, has very rich composition to suppress pro-inflammatory cytokines. On the other hand, this is one of my favorite slides actually, because we obtained the proteins from the EVPs or from the media, and then we looked in literature about the downregulated proteins in senescent cells, and we found about 130 proteins that are actually downregulated in senescent cells. So what does that mean? The media that is provided from stem cells is actually supplementing proteins that are otherwise downregulated in senescent cells. So this could be one way that the stem cell-derived media is supporting or antagonizing the senescence phenotype. Among these proteins, we find vimentin, NLRP3, and so on. We are in the process of validating these by Western blot analysis. We also performed RNA-seq analysis, and we got this beautiful volcano plot showing the downregulated mRNAs and the upregulated mRNAs after the exposure of WI38 cells after exposure to ionizing radiation to induce senescence. These are in the green part, the downregulated protein, the downregulated transcripts. Surprisingly, most of the downregulated ones are SASP-encoding mRNAs, indicating again or confirming the effect of the conditioned media on the SASP phenotype as we observed earlier. We're still looking at what the function of the upregulated proteins. We're looking at that. Collectively, the downregulated transcripts are basically, as you can see here, when we look at the function and the pathways, they are involved in chemokine, cytokine, array cytokine activities, and so on, indicating again that the conditioned media is actually suppressing the transcription or the transcripts that are encoding for SASP factors. On the other side here, I still observe a lot of extracellular matrix modulation, maybe indicating that the conditioned media is actually has some effect or modulating the extracellular matrix remodeling or tissue remodeling. This is another unbiased approach in which we measure the concentration of cytokines or measure the SASP factors or soluble proteins in the media that are in the media, and these are released from the senescent cells and also released from the cells that are exposed to conditioned media. What you see here, for example, these are the cells. They were exposed to ionizing radiation for 3 days to 7 days to 10 days as well. And here, these are the cells incubated with the human trophoblast stem cells. In all time points, we observe a significant downregulation of these cytokines or these soluble proteins or secretome that is coming from senescent cells as a result of incubation with the conditioned media. We also observe a reduction in IL-6 and IL-8 because, as I mentioned earlier, these cytokines are actually go in they have a different dynamics. They have different concentrations at different times. So here, after 3 days, we measured we saw a downregulation of IL-6. Here, also after 3 days of incubation with the conditioned media, 3 days post-IR, we found the reduction in IL-8. So all of these get together and tell us that the conditioned media is indeed suppressing the pro-inflammatory phenotype observed in senescent cells, or otherwise known as SASP. So now coming to the most important part and the most time-consuming part of this work, which is the isolation, purification, and quality control of assessing the quality control of extracellular vesicles or the exosomes. We isolated the EVs or the exosomes using size exclusion chromatography. We concentrated our EVs, performed electron microscopy, and as you can see here, the range of the size is between 30 and 100 nanometer. We also assessed the tetraspanin CD63 and CD63 and CD81, and observed that they are indeed present in our preparation. And this is the experiment. We treated cells with ionizing radiation 10 days after induction of senescence. These cells were indeed senescent. 8 days after exposure. And then here we added the EVs or the exosomes. And then we observed the effect on the SASP mRNAs at day 10. As you can see here, we observe a significant reduction in some of those cytokines that we measured at that time, and we are in the process to do a larger scale because we need more EVs or more exosomes to perform high throughput analysis. But as you can see here, IL-8, IL-18, CXL1, they are significantly downregulated upon the exposure or upon the treatment of the cells with the EV human trophoblast stem cell-derived extracellular vesicles. To take home a message, so the conditioned media, it did counteract the effect of counteract senescence. It promoted some proliferation. It counteracted the effect of the IR on DNA damage. It inhibited the effect of NF-κB, and also we observed less SASP production. When we characterized the media, we observed a rich composition of this media that suppresses the SASP, and also observed a number of proteins that are otherwise downregulated in senescent cells now being supplemented by the EVs. By the conditioned media from stem cells. Now, the recipient cells, we observed in RNA-seq and bioplex significant reduction in SASP, including mRNAs in the case of the RNA sequencing, and in the bioplex indicated that the proteins themselves are also downregulated in the media. This is basically the same that I just said. I would like to take you further to the in vivo experiment that we're planning to do, or is actually ongoing this week and next week. We're planning to inject the EVs, or the exosome, into aged mice and observe the aging markers. We also plan to inject the media as well. With that, I would like to thank Dr. Luigi Ferrugi for funding this work, the Office of Scientific Director. And I'd like to thank Miriam Gorospi and our lab. I'd like to thank Rafa, Isabel Bierman and Nate Price for their work on the in vivo work, helping us with the mouse work. I'd like to thank Exer Bioscience for being generous to give us the conditioned media to test it in our experiment. And if you ever need this kind of media or exosomes, please let me know. I'll be very happy to support you. I'm looking for more collaborations on the exosomes and and the age-related diseases as well. And thank you. Very happy.
Very interesting work. Yes, we'd really like to see where that goes. Who is the first up for questions? Here we go.
Of course, I'm going to ask about mitochondria. We look at mitophagy a lot, and we're also now starting to see more of mitochondrial-derived vesicles and signaling patterns that are coming from that. And we know there's some cross-interplay between mitochondria and senescence. Are you looking at that at all in your studies?
Actually, I tried to do this assay. It didn't go successfully, miserably failing. The thing is that I still have to optimize it because I need the certain number of cells in the well to do the seahorse, if you're familiar with that, of course. And it's been a little bit of a challenge, but we're optimizing these conditions to measure the mitochondrial activity, the oxygen consumption rate, and so on.
Excellent. We should talk afterward about potentially integrating your senescence markers with our MitoQC and MT-Chema mitophagy
Absolutely.
models.
Anyone else? Looks like that's it for now. Thanks so much, Kotb.
Thank you.